Hello everyone, and thank you for joining us today for this webinar titled Droplet-based single-cell analysis reveals hidden CD4 and CD8 CAR T-cell cytotoxicity. Before I introduce you to our speaker, I'd like to cover a couple of housekeeping items. You can submit questions to the speakers using the Q&A widget. These will be answered after the presentation. If you encounter any technical issues, you can click on the help icon for a list of solutions to common issues. And you can resize the webinar slides by clicking on the icons above the slides. I'm delighted to introduce our speaker today, Peter Mouritzen. Peter is the Chief Development Officer at Samplix, where he leads product and application development and brings over 20 years of experience in life science tools. Peter is an expert in single-cell biology technologies, droplet-based functional assays and assay development, and has collaborated with universities and hospitals to deploy single-cell functional assays. He previously held senior development roles in the life science technology sector, driving innovation at the intersection of molecular biology, instrumentation and applied research. Over to you, Peter.
Good morning, good afternoon, or good evening, wherever you are. I'm thrilled to be here today to be able to talk to you about functional analysis with single-cell resolution and how this is being explored for development of CAR T-cell therapy. Before I begin, let me just provide a brief outline of my talk today. So I thought I'd start with a technology introduction and then give you a couple of selected applications of how you can utilize a single cell platform like Xdrop for performing a functional analysis. And before really shifting to talking about today's main topic, which is CD4 and CD8 specific CAR T-cell cytotoxicity. And then finally, I'll wrap everything up in a brief summary. Just a little background about Samplix. Samplix was founded back in 2012. The company has more than 10 years of experience in microfluidics and droplet technologies. We are, as such, headquartered in Denmark and also have a hub in Boston to serve the American market. And other than that, Samplix is really about being able to support life sciences and medical research communities with microfluidic-based solutions. Really to deliver the highest resolution insights into functional cell biology.
Samplix offers three different categories of products. There's the instrument, which is the Xdrop. It's for droplet generation. It is a droplet generator, and it's a benchtop instrument. There are consumables, which are cartridges and reagents. The cartridge is what you see in the picture here. As you can see, there are 8 lanes, as we call them here, and each lane will load a sample. So you can process 8 samples in parallel. We also provide oil and other reagents for conducting various workflows. And then there are the digital tools, of which I would like to highlight the tool that you use for calculating cell number loading into droplets, which is very useful when you're doing cell-cell co-encapsulations to get the numbers right.
At the heart of the Xdrop technology is really the double emulsion droplets. Most people will know droplet technology from technologies provided, for example, by Bio-Rad for digital PCR, also maybe from 10x Genomics for doing single-cell transcriptomics. But here we are talking about double emulsion droplets. And so the difference really between the single and double emulsion droplets is that single emulsion droplets are small water droplets in an oil phase, whereas double emulsion droplets are water in oil in water. And so what you're looking at here is a cartoon of a small droplet. So this double ring here is trying to illustrate the oil shell, which is around the content of the droplet. And of course you will not load water in there. If you're culturing cells, you will load media. And you will do the same on the outside. And so this little container here is like a microreactor. So you can load it with cells, one or more. And the cells are supported in the droplet because there is media, of course, but also because there is a free diffusion of both CO2 and oxygen across the oil shell here. Whatever the cell secretes stays inside the droplet, and so this is a very good way to analyze secretion, because it's a very small volume. So it's only about 65 picoliters. So within a very short time, you'll be able to see accumulation of secreted compound to a level where you can actually detect it. So most assays performed on droplet technologies like this are very rapid. Other than that, of course, very important also for a cell-cell interaction assays, where you will bring cells in close contact with each other, and also be able to maintain cell pairs throughout your analysis. Another important feature is, of course, that we make 1 million of these per sample. So you get 1 million small microreactors, which you can carry out experiments in. And so the droplets are very monodisperse, and they are compatible with flow cytometers and cell sorters, and handling likewise is just by standard pipetting. They are incubated in standard CO2 incubators and so on. So it very much plugs right into the existing laboratory infrastructure.
A very generalized workflow for a single cell assay would look somewhat like this. So it starts by 3 simple pipetting steps. You add oil to the cartridge, you add your reaction mix containing your sample, yourself, and then outer buffer that goes outside the droplets. Then you pop this cartridge into the instrument front drawer here and press start, and then 45 minutes later you'll have the cartridge out again, and you can harvest all the formed droplets from this reservoir here and put them into a culture tube, which you then put into an incubator, standard CO2 incubator, and then you incubate. And once you have incubated, you're ready to do your cell sorting or flow cytometry to analyze.
Okay, let me shift gear and talk a little bit about how can you really utilize a technology like this for and that quite high screening capability for something very useful. So let me start with an assay that we have been working with for some time now. And so we're actually quite fond of an assay from Italy where we can perform this in bulk, of course. And the way the assay work is by anchoring catch antibodies on the surface of the effector cells. And those catch antibodies recognize the cytokine that you're interested in. You then incubate the cells, and then cytokine secreted will basically bind, of course, to these catch antibodies on the surface of that effector cell. And then you have detection antibodies that will also recognize the cytokine and bind to this. In an assay like this, of course, you will have many different cells. Some will be high secretors and others will be low secretors. But you can, of course, imagine that if a high secretor is next to a low secretor, there'll be some transfer of cytokine from one cell to another. And so when you analyze something like this, here we have done it for a TNF-alpha secretion by T cells in a PBMC sample. You can see that if we have not activated the T cells, we get this kind of profile of cytokine expression on the flow cytometer. And if we activate the cells, it looks like we are really pushing everything up in cytokine signal, and it looks like this. Now, if we are performing this assay as a single cell assay, so now we are really putting the cells inside droplets, we're preventing this crosstalk that we saw in the bulk assay. And so when we now do this type of analysis, we can see that indeed there are some cells that start secreting a lot of cytokine upon activation, but it's by far the majority of the cells. The majority of cells don't change much profile, but there's definitely a small fraction of very high secretors that are revealed by the cytokine assay.
Another important assay is the single-cell cytotoxicity assay, or the cell-killing assay. I'll be talking about this assay basically for the remainder of my talk, so I just want to spend a little time to explain it in carefully. So before we conduct the encapsulation, we take target and effector cells and stain them. The K562 cells were used in this example, and NK cells also. They're stained, and then they are encapsulated in droplets, and after this they're incubated. And so the encapsulation itself produces different types of droplets. There are both empty droplets, there are co-encapsulated droplets, and then there are droplets that only contain one of these two cell types. And these actually serve as a nice internal control, because looking at a droplet like this, you will really be able to tell what is the amount of cell death in droplets that only has the target cell and not an effector cell. And to monitor cell death or apoptosis, we either use a caspase 3,7 substrate or propylium iodide, both work. And to monitor whether the effector cell has recognized that there's a target cell present, we include a granzyme B substrate, which will convert into a Fitch signal that makes the entire droplet light up if that happens.
In real life, the assay looks like this. Over here you can see the very monodispersed droplets with cells inside, and down here you can see the granzyme B signal in some of these droplets. We're looking at droplets that has been incubated for 4 hours, and they are with IL-15 stimulated NK cells. Up here you can see the an enlarged droplet, where we can see there's a green droplet, of course, and so we can see there's both the violet stained NK cells, the red stained K562 cells, and death is indicated in the K562 cells by the propylium iodide that we're using in this example here. Flow cytometry is really our go-to workhorse when we have to quantify this, and so let us first have a look at the K562 droplets, which are here in this quadrant. So they are positive for the red dye, and so there is very little granzyme B signal developing over these 4 hours in these droplets, whether with or without IL-15. In case of the NK cells, there is a bit more signal developing in case of IL-15 stimulating than without, which makes sense, I think. And in particular, after 4 hours, there's about 14%. So the really interesting one is, of course, up here. So here we have the droplets co-encapsulated with both cell types. And here are both the granzyme B signal quantified and droplets which has both granzyme B and cell death indicated. So after 4 hours, you can see that we have you have to kind of put these two bars together to know the full granzyme B signal. So we are well above 40% of droplets that are granzyme B positive after 4 hours, and you also have 20% of them being containing dead cells. I think it's important also to note that already after 30 minutes, you have a significant amount of positive granzyme B cells with this assay. It's very sensitive, actually. So researchers at the National Center for Cancer Immunotherapy at Herlev Hospital have utilized this assay to sort out killers and non-killers from tumor-infiltrating lymphocytes and characterized these in terms of expression profile utilizing the 10x Genomics platform. And so here we're looking at a UMAP plot of both killers and non-killers. And so the shape of this plot is, of course, based on the expression of gene expression within each of these individual cells. And this is why it looks like it does. But when we overlay this plot with the information about which cell was a killer and which was a non-killer, there's a clear pattern, and obviously very interesting to understand what happens in places like this and here. Of course, this is also a way to quickly identify TCRs. We're sorting out the killers, and so in the killer fraction we can look for the TCRs, and so we find more than 200 different TCRs, but there are some that are very frequent. This particular TCR number 1 here is making up 7%, and the first 15 TCRs is making up 45% altogether. All right, let us get to talk about the case study about revealing hidden CD4 and CD8 CAR T-cell cytotoxicity.
At this point, I really need to acknowledge the team at Odense University Hospital, the Department of Clinical Immunology, and in particular Silvera, who's been instrumental for this work, also Mike, who's heading research there, and Tom Barrington, who is also acting professor at the university. I also want to highlight a paper that the team had out last year in JOVA, which is like a video paper, but there's also a written script. It's on the droplet-based cell cytotoxicity assays applied in CAR T cells. The team at Odense University Hospital is working on development of new CAR T cell therapies, and they're particularly interested in coping with the situation where the tumor undergoes an antigen escape, where it simply loses its antigen, which the CAR recognizes. And for this purpose, they are looking into different targets for the CARs, and they're also looking into constructing dual target CARs. The classical, the traditional CAR, the first CAR that a patient was ever treated with was the anti-CD19 CAR, and it has proven to be very efficient, but nevertheless there are situations where the patient's tumor loses the CD19 antigen, after which the CAR T cells are no longer able to control the development of the tumor. And for this purpose, they have also looked into the B-cell activating factor receptor BAFF-R as a target. The BAFF-R is a TNF receptor superfamily member. It's crucial for B-cell survival through its binding of the soluble ligand B-cell activating factor BAFF. And because of its role and being so crucial, it is appearing as a quite promising target. And so they're also looking into the dual target CARs, where they're combining these two, and this can be done in different ways. As you can see on the drawing here, there's an aim B and an aim C, where there's a different orientation of these two recognizing units, and then there is an aim E, which I'm only showing the rough outline of. This will be revealed later, but one of the aims with this particular construct is also to improve the durability, to avoid exhaustion of the CAR T cells over a longer time. Anyway, so these are the constructs that they have been using to make CAR T cells. And the first experiment to do when you do this is, of course, to monitor how cytotoxic is this. And for this, they did bulk cytotoxicity assays, where they incubated the CAR T cells together with the ECO1 cells in bulk, and then after some time they analyzed on flow cytometry for dead cells. And so here on the y-axis we have percent dead target cells, and so here for the first three ones, which are the monospecific CARs and the AB CAR, it's a very efficient control of the target cells. The ANC is performing a little less well, and AME is also looking quite well. And then this can be compared here to the untransduced cells, which is in this range here, and here, just for reference, is the death of the target cell only. When the ratio of target and effector cells are changed, so there are now more and more target cells, the killing also drops. But the pattern is the same. It's C and is performing less well throughout the study. Because the overall aim of this study is really to be able to develop CARs that are able to deal with the antigen escape situation, then went ahead and created knockouts mimicking the loss of CD19 by doing making a CD19 knockout, and also mimicking loss of BAFF-R. And then they conducted the bulk cytotoxicity assays again. And of course, now when you have knocked out the CD19, the anti-CD19 CAR is not performing well. The BAFF-R is performing as before, and B is also looking good. C is still looking poor, as also in the previous study on the wild-type geckos. E is still looking fair, and also you can see the same kind of like trend as you move to different target effector ratios, as shown in the graph here. For the situation where we are mimicking loss of BAFF-R, it's the vice versa situation. Now it's the anti-BAFF-R that is not performing well, whereas the other ones are looking fair. Maybe C is actually looking a little bit better here, but then it drops in the next target effector cell ratio. So overall, it is again E that is looking promising of the dual target CARs. Of course, the really interesting question is how are these different CARs performing in vivo, and for this reason the team went ahead to conduct mouse experiments. The mouse were simply injected with wild-type gecko cells carrying a bioluminescent reporter, and this was done on day 0, and then following on day 9 they were injected with either untransduced cells or with the different CARs. And then they were followed up to 62 days, and immediately it's evident, of course, that the UTDs are not really able to control the spread of the tumor inside the mice, but the anti-CD19 CARs are doing fairly good during this time. A and B is also looking good. MC is again not looking very good. It's not able to control it very well, the spread of the tumor, whereas ME is kind of like an in-between intermediate result. So here we are performing the experiment with the wild-type gecko cells, but what we're really trying to get to is, of course, how do they perform in the situation where you have antigen loss, antigen escape, and so this was performed in the next experiment.
The team then went on to build 2 new gecko cells, a BAFF-R knockout with a red fluorescence protein, and a CD19 knockout with a green fluorescence luciferase reporter. And these were mixed 1-to-1 before injection. And so now you basically have the situation where you have antigen escape. And so now we're trying to follow how does that look when we look only in the channel for the BAFF-R knockout, and if we look in the channel, the green fluorescence luciferase for the CD19 knockout. So it's evident just from these images here that the BAFF-R is probably a very important parameter for the gecko cell, for the B cell survival in these mice, because they do not grow very well compared to the CD19 knockout gecko cells over here. And so therefore the results over here is a little bit more difficult to interpret. There is a small diagram down here that allows you to see that the UTDs definitely probably perform, or probably there's more tumor growth in the UTDs here after 19 days, and you could probably say something similar about the BAFF-R CAR T cells. So now and that is also expected because now the BAFF-R has been knocked out. So nothing unexpected there. It's hard to say much about the rest. It's a more clear picture over here in the CD19 knockout mice gecko cells in mice. So now we have escape in the anti-CD19 CARs. And we have a little bit of escape also here, or at least there's some tumor growth in B. and by far is doing a pretty good job, and that's also what's reflected down here. Here's the by far, and here's the AME down on the axis here, very little signal.
At this stage, the team also wanted to investigate whether some of these observed differences between the different cars could potentially be explained by the higher resolution present in the droplet single-cell assays. And so for that reason, they carried out experiments like the one shown here, where they encapsulated wild-type Jaeger cells together with CAR T-cells in the assay that we have already discussed. Did that together with the grandson B substrate and the caspase-3 so we're able to monitor both activation of the effector cells, of the CAR T cells, and also death of the tumor cells. And so this is what it looks like. Here we have the co-encapsulated cells in droplets. Some turn up very green here, as you can see, and down here you can see the CAR T cells violet, Jaeger cells in far red, clearly in this side inside this droplet. And the flow cytometry, of course, mostly interesting here to look at the co-encapsulations. So I'll focus on this. And so some results down here looking at both the BAFF-R CARs and the anti-CD19 CARs. And first of all, again, as previously observed, only about 10 to 20% of the monospecific CARs T cells are really exhibiting a fast killing, and here we're talking about 6 hours. So it's not when you're working with these populations that you'll just have a 100% activity. That's not the case. And that's what the single cell assay is able to reveal. But I think more important here is that we can see there's a clear difference between the BAFF-R in terms both of the grandson B secretion relative to the anti-CD19 CAR. And there's also a difference between these two CARs in terms of the cell death that was monitored, the caspase-3 activity. Before proceeding with the analysis of the other CAR constructs, the team was interested to increase the resolution of the assay by introducing 2 additional stains, one for CD4 and one for CD8 T-cells or CAR T-cells. And while this may seem relatively simple, it does, in fact, of course, increase the number of colors in the assays from 4 to 5, and that did turn out to have some challenges. At this point, we became aware of the StarBright dyes, and the ability to stain CD4 and CD8 became an opportunity here that we had to pursue. Choosing the StarBright dyes, we can still keep our grandson B substrate here and our caspase-3 substrate over here, as well as the other cell stains that we're using in the assay. And so it's really about both, having the Stoke shift, the large Stoke shift of these dyes, but also the brightness of them that allows us to utilize this. And so this is demonstrated down here where we have stained human PBMCs, where we can see very bright signal here from CD4 and from CD8 over here. And also when we do flow cytometry, we're able to gate out and identify based on these antibody stains, StarBright antibody stains. The last test to do before performing the real experiment was a test where we tried to make sure that none of these antibodies would interfere with the functionality of the CAR T cells. And for this reason, we did a small experiment where we either stained in this line here with anti-CD4, anti-CD8 StarBright dyes, or we didn't do it. And then we looked in the 4 different droplet populations, the Jaegers alone, the co-encapsulations, and so on. And we're able to see that we obtain more or less the same pattern and the same percentages. Those are hard to see. But down here you have kind of like the quantification in diagrams, and you can see the grandson B activity is very similar in these co-encapsulations between the cells that are not stained and the cells that are stained. And the same goes for the caspase-3 activity, very similar signals. With the absence of interference affirmed, the team went ahead to test both the monospecific CARs, represented here by the anti-CD19 CARs, and also some of the dual targeting CARs, B and AIM-E. And if we focus first on the anti-CD19 CARs, it's not surprising that the CD8 positive cells are both cytotoxic. They have grandson B activity, and you can also definitely see that there is caspase 3-7 activity in those co-encapsulated droplets. And this is in contrast to droplets where you don't have any Jaeger cells present. So you're looking at this difference here, really. What's interesting to see is that there's actually also both grandson B activity and also a certain level of caspase 3 activity in droplets with CD4 positive cells. On the dual targeting CARs, there is more or less a similar result, possibly a little bit better performance of the AIM-E here we're just looking at grandson B activity, but there's a little higher activity. Let me try to summarize over these last few slides. The question is really, does the single cell cytotoxicity align well with in vivo data? And if we start out by taking a look at the dual targeting CAR, it was evident from the in vivo experiments that the AIM-E CARs were able to control the proliferation of tumors better than the AIM-E CARs. And if we take a look at the corresponding results from the single cell cytotoxicity assay in droplets, we could not see a huge difference between these two constructs, but there was a tendency that the AIM-E CARs performed a bit better. On the other hand, if we took a look at the single monospecific CAR constructs, looking at anti-CD19 versus anti-BAFF-R, there was a clear difference. The anti-CD19 CARs were more cytotoxic than the anti-BAFF-R cells, and we could monitor both higher grandson B secretion and also target cell killing. In the in vivo experiments, this was a little hard to conclude, in particular because this part of the experiments was difficult to evaluate because the BAFF-R knockout Jaeger cells did not establish themselves well and proliferate in the mice. But in correspondence with Silveras, he said, yes, unfortunately, the anti-BAFF-R CAR T cells are performing less well in all studies compared to anti-CD19 CARs. In vitro, they are very similar. The difference is only in vivo. Okay, let me make a brief summary. So Xdrop is really a tool that you can use to carry out functional assays at a single cell resolution. It's a high throughput tool that will allow you to screen millions of cells, and subsequently you're able to isolate viable cells, and they are subjectable to 10x genomics or other downstream applications. The platform is really easy to use, and the droplets themselves, importantly, are compatible with both flow cytometry and cell sorters. On the Bio-Rad StarBright stains, I would like to highlight that this has really enabled more complex assays on our platform. I would like to just draw your attention to a number of publications that are available from Samplix homepage, both on the cell biology topics that we discussed here today, but also on genomics applications. So with this, I just want to thank you for your attention, and over to you, Mike.
Before we go into the Q&A, I would just like to take the opportunity to give a little bit more information on the StarBright Dye portfolio and StarBright Dyes which were used in today's webinar. Bio-Rad has a broad portfolio of antibodies validated for Western blot, microscopy and ELISA in addition to flow cytometry. StarBright Dyes are a range of 36 superior dyes excitable by the 5 common lasers typically found in flow cytometers. 32 of these were designed on the ZE5, a traditional flow cytometer. They also work in spectral flow, and 4 specifically for spectral flow designed on the Cytec Aurora. They are bright with narrow excitation and emission profiles, require no special buffer when multiplexing, are fixable, and can be pre-mixed and stored prior to use. We have over 1,900 conjugates available as 100 test sizes and are continuously adding more. We also have CD4 conjugates available in 25 test sizes, making it a convenient and cost-effective way of evaluating StarBright dyes on your instrument. Despite having a broad portfolio, we know that the right fluorophore is not always available on your antibody of choice, so we have developed a StarBright dye labeling kit. This labeling kit consists of 2 parts. The first part, adding a SPI tag to the antibody. Once the antibody is conjugation ready, you simply incubate with the SPI tag labeled StarBright dye, and an irreversible covalent bond is formed, resulting in stable, labeled antibodies. This simple protocol results in highly stable conjugates with similar performances to directly conjugated antibodies, examples of which can be seen in the images. This is available for 32 StarBright dyes, significantly expanding your fluorophore options, particularly for the 355 and 405 nanometer lasers. They're compatible with most antibody types and work in most buffers, even those containing BSA. In summary, StarBright dyes allow optimal panel design of any size by providing choice and flexibility when multiplexing. If you do not have the format you require, our Trailblazer labeling kit allows you to conjugate primaries and secondaries with 32 premium dyes. No special buffer is required, removing the extra cost, and they can be fixed in both formaldehyde and alcohol-based fixatives. The brightness with improved spectral properties mean there is less spread, leading to high cell resolution when multiplexing. Finally, the ability to pre-mix and store means preparation time is reduced and less error between experiments. Go to bioradantibodies.com/StarBright to find out more.